Hi, I'm Greg Capareso, and I'm going to be leading the next section of the tutorial, which is focused on longitudinal microbiome analysis. And so I'm going to scroll down to the downstream tutorial section and click on longitudinal microbiome analysis. Now, over the last few sections, we have done some longitudinal microbiome analysis. This section differs in that we're going to be focusing on using the Q2 longitudinal plugin. This provides some specific actions that are helpful in longitudinal microbiome analysis. And in particular, we are going to work with some of the visualizations in this section that allow us to track visually features of the microbiome that are changing over time. Now, just like we did for some of the other stages, we're going to take a few steps here to prepare our feature table for longitudinal analysis. These preparation steps are things that you may occasionally want to do for other types of analyses, and they're also not essential that you do. They're not essential to do for this type of an analysis, but they will help us speed up some of these analysis steps and focus in on some taxa that are most likely to be relevant here, and some other features that are most likely to be relevant here. So the first thing that we're going to do is we're going to take the feature table that we've been working, which, as I'm sure you remember, has amplicon sequence variants as its features. And we are going to collapse those features into bacterial genera based on the taxonomic assignments that we made earlier in the workshop. And so this is going to provide a couple of convenient features. So first of all, because there will be fewer bacterial genera than amplicon sequence variants, because we're grouping sequence variants or ASVs together into genuses, we're going to end up with fewer features than if we worked with the full amplicon sequence variant table. The other convenient feature of this is that when we look at the resulting visualization, rather than see those opaque feature identifiers showing up, we're going to see names of bacterial genera showing up. And so that's helpful for interpreting the results. Now, there is a downside to this. The downside is because we're collapsing sequence variants into genera, we're effectively decreasing the resolution of our analysis. And so, for example, if two ASVs in the same genus illustrated different temporal properties, we would no longer have the ability to see those differences because those two ASVs are getting grouped into one single feature. But so, sometimes you may want to do this, other times you may not want to do this. But here we're going to take these steps, and it'll at least give you an idea of how this type of collapsing might be used in some of your analyses. Okay, so I'm going to switch over to my Galaxy server now. And the first thing I'm going to do is I'm going to search for this tool that I'm interested in. And so it was called Taxa Collapse. And so if I search for it there, I can see here's Chen 2 Taxa Collapse. And so I'm going to click on that so that I get this tool pulled up. Now, I currently have fewer items in my history than you do. And so you'll see that my table field is being populated with the one relevant type that I have here, which happens to be associated with filtered table 4.qza. You should go ahead and select filtered table 4.qza from your dropdown. Similarly, I only have taxonomy.qza. That's the only relevant file that I have for this field. That is probably the case for you as well, so you can just go ahead and select that. And then I see that there is one required field here, and that is level. And so what that is it's the taxonomic level at which the feature should be collapsed. Now, this is going to be based on the taxonomy that you're using. But for us, for green genes, the 6th taxonomic level corresponds to genus. And so if I just go back to my tutorial, I can confirm that it's suggesting that I set level to 6. And so I'm going to go ahead and do that, and then I'm going to click the execute button. Now that'll take just a minute to run. And while that's running, I'm going to come back over here and I am going to select the name that I'm going to save this history file, this file from my history as. And so I'm just going to copy that name. I'm going to come back over. I see Galaxy has finished running. And as you've now done many times, I am going to come in here and I am going to edit the name attribute of this entry in my history. And then I'm going to hit save. So now you see I have genustable.qza showing up. And so I'm going to come back to my tutorial and scroll down to this next section. And so you may think at this point that it is not possible that there are filtering methods in QIIME 2 that we haven't yet encountered in that workshop, but you would be wrong. We're going to use a new to us filtering method here, which is called filter features conditionally. This is in the feature table plugin. Now, the way this works is it uses both abundance and prevalence for filtering. And so prevalence refers to the number or the fraction of samples that a feature shows up in. And abundance refers to the frequency of a feature within a single sample. And so this action is a conditional filter because it integrates both abundance and prevalence. And so if I follow the guidelines here, so if I set prevalence to 0.1 and abundance to 0.01, that corresponds to a 10% prevalence and a 1% abundance. And so the way that this filter works is it will retain features that have an abundance of at least 1% in 10% of the samples. And so that is a lot to follow there. But again, it is going to keep features that have an abundance of at least 1% in at least 10% of the samples. And so what this is going to allow us to do is focus on the features that have relatively higher abundance across more of our samples. And so it is just allowing us to selectively focus in on some genera of interest. Now, this is not something that is required, but it does help this analysis run more quickly. If you are running this on your own data set, you might want to apply some less stringent filtering here, or at least experiment with those values. We're optimizing in this tutorial for something that's going to run relatively quickly. So let's go ahead and run this next command. And so I'm going to go ahead and just copy this name, filter features conditionally, so that I can use it in this search field up here. You can see that's the first thing that shows up. And it has selected the correct artifact here. And so it has selected genustable.qza. Now, we said our abundance was going to be 0.01 and our prevalence was going to be 0.1. So I'm going to go ahead and set those, click execute, and give this a minute to filter. Again, while that's filtering, I'm going to come over here and I'm going to copy the new name for this feature table. We can see that that is still running, so we'll just give that a minute. Oh, and there we go. So once that's done running, I'm going to click Edit. I'm going to come up here and I'm going to paste that new file name that I copied, and I'm going to save that. So I now have an artifact called featured genus table.qza. Now, the last thing that we're going to do to prepare for these next analysis steps is we are going to convert our feature table from frequency or raw counts to relative frequency. And so that is a relatively simple computation that is performed on each sample individually. And so simply what's happening there is the count of each feature is being divided by the total feature count across all features in that sample. And so it's basically just taking each per sample genus count and converting that from a raw count to a percentage of the sample that is composed of that feature. And so we're going to apply this to our filtered genus table.qza. And there's no parameters that we need to set here. And so this one was called relative frequency. When I search for it, again, it's the first one that pops up. Again, it's taking my filtered genus table as input. No parameters here, so I'm just going to call execute. And again, while that's running, I'm going to go back to my tutorial and I'm going to copy the name that we are going to save this as. Looks like this is still running and just completed. So I'm going to go ahead and edit attributes. I'm going to edit the name and put in genus-rf-table.qza, and I'm going to save that artifact. Okay, so now we're ready to start generating some of these plots. And you can see there's some more options that we're going to set here. The first of these plots that we're going to generate is called a volatility plot. What this does is it allows us to track features of the microbiome, things like alpha diversity metrics, across time. And so what we need to do here is we need to provide the feature table that we want to work with and the data that we want to plot across time. And actually, in this case, the feature table is actually optional. It's really the metadata that is the focus of this plot, including metadata that we will be reading from QIIME2 artifacts. And so I'm going to end up flipping back and forth between these two documents. But the first thing I'm going to do is I'm going to pull this action up, so volatility. You can see it's pulling up feature volatility and volatility. We're going to look at volatility, not feature volatility yet. And we're going to start with metadata. And so this first entry is populated as I need it, so metadata from TSV, and then sample metadata.tsv is the input that we're providing there. Now, next what we're going to do is use several QIIME2 artifacts as metadata here. And so the artifacts that we're going to use is the unweighted unifrac UMAP matrix. And so that's going to be the two UMAP axes that we were visualizing in the previous plot. And so when we use that as metadata, the value that is associated with each sample would simply be the placement of each sample on each axis. And so for UMAP, there's going to be two axes. And so each sample is going to have a UMAP axis 1 value and a UMAP axis 2 value. We will also look at Faith's phylogenetic diversity here, evenness, and Shannon entropy or Shannon index. Okay, so I'm going to go ahead and I'm going to add each of these. So insert metadata. I'm going to select metadata from artifact. And I will start with UMAP. That's my unweighted unifrac UMAP. I'm going to do this again. So added another metadata. And then this time we will do faith's PD vector. So that's faith's phylogenetic diversity. We'll do another one. And we're going to do metadata from artifact. And we will choose Shannon. So Shannon vector.qza. And then one last time, we're going to do metadata from artifact. And we are going to select, which one did I not do yet? Evenness, I think. And so evenness, Shannon, faith PD, UMAP. Okay, so now let's refer back to the tutorial for the next steps. And so the next thing that we're going to do is we're going to set the state column to weak relative to HCT. And so just to make sure you don't have any typos in that, I recommend that you copy this text. So select it, choose copy, go over to Galaxy, paste it. And so I did Command V, but you can also edit, paste. And then I just kind of visually inspect it to make sure I copied the whole thing. Now, if I come back here, you can see that in this case, there are some additional options. And so we're going to expand additional options. And so click here for additional options. We can add a feature table. Notice that this is a relative frequency feature table. And so that's the one that we just generated, the genus RF table. You can see that is probably the only one of that type that you have. We need to set the individual ID column. And so we are going to set that by saying provide a, oh, sorry, I'm in the wrong spot. So individual ID column, we're going to say provide a value. And then I'm going to paste in patient ID, which I copied from the tutorial. And then the last thing is we're going to set a default group column, and we're going to make that auto FMT group. Again, I recommend copy and paste here. And so I had already selected provide a value, and I'm going to paste in auto FMT group. Now I'll go back and I'll make sure nothing else to set. Press the execute button. And so I will come in here, I will scroll down, and I will click execute. So that's going to take just a minute to run. Again, while that's running, I'm going to come over here and I am going to copy the name that was assigned to this. When I come back, I can see that that has completed, so I can edit and assign a new name and click save. So let's take a minute now and take a look at this plot. And so if I click on the name of that in my history, I'm sure you're all experts at this now. You can then scroll down and you can click View at Chime2 View. That's going to open in a new browser tab, and you should see a plot that looks like this open up on your screen. Now, one thing you'll notice in these plots, and we're going to look at a few of these, so I'm going to spend a few extra minutes on this one right now, just to familiarize you with what is being shown here and what controls you have available to you. So the x-axis here is weak relative to HCT. If you go back to the tutorial document, you can see that that is the state column that was required. And so the reason that that's required is because that becomes the x-axis of this plot. The y-axis here is the metric column, and that is something that you get to choose within this visualization. And so all relevant metadata will be available for you to view here. And we will, so some of the things that we can do here is, like, right now we can see that we're plotting day relative to FMT, which is why you see this really nice, linear association here, because obviously the day relative to FMT is somewhat correlated with the week relative to HCT. But probably more interesting would be to, for example, look at something like Faith's phylogenetic diversity. And so here now we are getting a view of Faith's PD, which, as a reminder, is a metric of community richness. And we're tracking that across the week relative to HCT. Now, you probably have also noticed the different colors that we have in this plot, which represent different groupings of the sample. Now, this auto-FMT group, you may recognize because this is one of the additional options that we set here. And so we have a default group column, and that was auto-FMT group. You can choose others. And so, for example, if you wanted to look at this on a per patient basis, you could group the samples based on their patient. And then actually what you end up with here is something that looks a lot like the ordination plots with time axes that we were looking at in the last section of the workshop. But here we're going to focus on grouping these based on auto-FMT group. And so what that means is that the individuals who received the auto-FMT treatment are showing up in blue, and the individuals who were in that control group, so they did not receive the FMT treatment, are showing up in orange. And so what I think is interesting here is, so this plot, again, is timed relative to when they received their HCT, so their stem cell transplant. So that is week 0 here. Now, anywhere from about a week to 3 weeks later, 4 weeks later, these individuals received their fecal microbiota transplant or not, depending on whether they were in the treatment or the control group. And so what we can see is, starting around 3 weeks after treatment, we begin to see a separation of these groups. And so while these were similar or maybe even reversed in their richness earlier, we can see that after the treatment, their microbiome richness is starting to increase. Now, that is in contrast to what was happening just before and during the treatment, where their richness was decreasing. And that is typically frequently what happens when you take antibiotics, as you would expect. And so while the HCT group, the control group here may not really be changing, so they maybe are looking more or less stable over time, we're seeing that the control group starts to increase in their community richness. Now, this is not a statistical interpretation necessarily. This is just a visual interpretation of the data. If you wanted to quantify this statistically, you would use most likely some of the LME techniques that were covered in the alpha diversity section of the tutorial. But typically, it's important to get both the statistical perspective of the data and the visual perspective of the data. Without having both of those, you could end up misinterpreting what, for example, a statistical result is telling you, or what a visual result is telling you. Now, you do have some other controls in here, which I encourage you to play around with. Of course, you can change color schemes if you want. You can do things like add error bars to these plots. You can change the mean line, and so that you can see is the mean across each of these groups. And then you can also see that you can change the width of what we call the spaghetti lines. And so the spaghetti lines are tracking the individual patients in the background here. And so if we increase those, you can see that those will either decrease or increase in width. You can change the opacity of those. Similarly, opacity of the mean line. If this is less useful with bigger data sets, but you can look at individual points for all of these. So for the mean, you can add points here. You could add points for the spaghetti lines in the background. I tend to leave those off just because they kind of get in the way when you're looking at a lot of data. Okay, so that is what we call a volatility plot. I am going to select a few other columns here. And so, for example, if we look at Shen and entropy, we see a similar trajectory here where we get this decrease before and during the treatment, and then the treatment group begins to increase in their community richness a few weeks after the HCT. We can also look at the UMAP axis 1 value. And so what I did there was I just selected axis 1 as my metric. And so now this would actually be basically the same data that we looked at when we did the ordination plots with a custom axis. And so this would be axis 1 of UMAP here. And then this axis, of course, is time. So let's go back to our tutorial now, and we're going to generate another one of these plots. But this time, rather than doing this based on the week relative to HCT, we're going to do this on the week relative to FMT. And so we're going to, I'm going to go ahead, I just copied week relative to FMT, so I have that. And one feature that is kind of handy here, oops, that was not what I meant to do, is if I select run this job again, so I still have this expanded, I can do run this job again. What this is going to do is it's going to give me the exact job that I ran there with all of the parameters that I filled in. But what I can do now is I can edit just the one that I want to change. And so I'm changing that to week relative to FMT. I'm just going to confirm that the additional options are the same. So I still have my genus RF table as input, my patient ID, my auto FMT group. And so that all looks good. So I'm going to go ahead and execute that. And so that can be handy in cases like this where you have a lot of different parameters that you set, and maybe you don't necessarily want to just go through and set all of those again just to make one little change in the inputs. So I'm going to come down here and I am going to copy the name, the desired name of this output, this completed running, so I can go ahead and edit my attributes, and I'm going to call that volatility plot 2.qzb. And so now I see volatility plot Q. I am going to, whoops, I keep doing that. I'm going to click on that, and then I'm going to go ahead and view at Chime 2 view. Now, this is the same thing that we were looking at before, except we've shifted the 0 here. And so the 0 now, instead of being weeks relative to HCT, is going to be week relative to FMT. And so go ahead and select Faith's phylogenetic diversity again. So get a plot of Faith's phylogenetic diversity by week relative to FMT. And you'll do that using the controls in this plot. Okay, so you should have that up by now. What you probably did was go to metric column and then find Faith PD in there. And you'll see that we have Faith's phylogenetic diversity. Now, this is interesting. So like we see our 0 here. In this case, I suspect just due to chance, we're seeing that there is some separation of those before the treatment. But at time 0, this is when they have received their fecal microbiota transplant. And so what you can see is like when you're looking at like around week -1 or week -2, there's some instability here. You see the orange and the blue spaghetti lines in the back crossing each other quite a bit. But once you get past week 0, so once they have received the fecal microbiota transplant, you can see that these groups are separate. And they are pretty distinct from one another at this point, at least until we get out to about week 3. Now, that might be a little concerning, so you might be wondering, oh, well, is this only a temporary effect of the FMT? But what you should notice here is that at this point, we have very few samples remaining from these, or we have very few individuals who are still providing samples. And you can tell that by the number of spaghetti lines that are showing up in the background here. And so like there's only a couple of oranges or maybe 1 or 2 oranges that are showing up at these later time points. Like it looks like there's 1 individual who has a week 5 sample and 1 individual has a week 6 sample. And so really around week 3 is where we stop having sufficient data for this analysis step. We can turn on some error bars here just for fun, just to see like how they are separating from one another. But again, this sort of supports what we were hoping to see with this data, and that is that the fecal transplant has an impact on the microbiota, and specifically in this case, it is restoring some of the richness that had been lost in the lead up to the stem cell transplant. We can explore this in the context of some of the other alpha diversity metrics, and so Shannon entropy. We can look at evenness. We see similar trends with both of these. We can look at axis 1 of UMAP, and we can see that there's a separation in the microbiome composition on axis 1, and we'll see so not so much on axis 2, but that is showing up probably because that's one of the main driving factors here is this separation. And so recall that with ordination methods, purposefully the axes are not correlated with one another. And so it's not surprising that if we see what we're looking for on one of the axes, we won't see it on another one of the axes. Now, you might also notice here that we have lists of bacterial genera. And so this is what we got by providing that genus table as an optional input. And so what I can do is I can sort through some of these and I can track how these individual genera were changing with time relative to the FMT in these two different groups. And so I'm selecting these more or less at random. You can see, though, that if you have a lot of these, this can be pretty challenging. So, for example, to identify which are the more relevant ones. But luckily, Chime 2 and the Q2 Longitudinal plugin specifically have a tool that helps you with that step. And that tool is called the feature volatility plot. And so that's what we're going to generate next. And so feature volatility, like you may be able to tell from the name, is tracking volatility of the features in the provided feature table over time. And so here you can see we're going to, we have somewhat of a complex setup again, because we want to integrate all of these other bits of data as input. And so let's go ahead and run this command. And so this command is called feature volatility. I'm going to copy that so I can use it in the search box up here. And you can see now feature volatility is the first one that's showing up here. So unlike volatility, feature volatility requires a feature table. And in this case, it requires a feature table frequency, so not that relative frequency table that we created. And so the one that we're going to provide here is filtered genus table.qza. And then let's refer back to figure out what metadata we're going to include. So again, we're going to include our sample metadata. That's metadata that's coming from a TSV. And then we're going to include those same artifacts as metadata that we used for the regular volatility plot. And so I'm going to come in here, metadata from TSV, sample metadata.tsv. That all looks good. Now I'm going to insert metadata and start inserting these metadata that I have from artifacts. And so I'll do UMAP first. Then I will do the, where did it go? Faith PD vector. Then I will do the Shannon vector. And then finally, I'll do the evenness vector. And so that I should now have, let's see, so I've got 5 metadata entries here, 1 from a TSV and 4 from artifacts. Now we have a little bit of additional information, and so state column, we're going to set to week relative to HCT. And so state column, week relative to HCT. And then under additional options, we're going to do individual ID column to patient ID. And so I'm just going to copy that to make sure I don't make a typo. Click here for additional options. And then that was that individual ID column. Okay, so I'm going to go ahead and hit execute. And then while that's running, I'm going to talk a little bit about what this plot is doing. Okay, so this is going to generate a few different outputs. The most relevant one for us is going to be the volatility plot.qzb. And so what's happening behind the scenes, what the plugin is doing right now, is it is running a supervised classification analysis to identify which features provide the most predictive value for the time variable here. And it uses that, not, of course, because we're trying to predict time, because we already know time for each sample, but we're using that to try and identify which are the most relevant features. And so if there are features that are most consistently changing with time, then those will be identified as the most important features in this analysis. And so that step completed running. And so 111 is the result that I'm interested in here. So this is Chang-2 longitudinal. And this is our volatility plot.qzb. And so if I click on that, I can view that at Chang-2 view. Now this time I didn't select a default group column. And so what this is doing is it's grouping all of the samples by the patient ID. And it is showing the abundance of the feature represented by metric column across time. And this is going to be the feature that it starts with is going to be the feature that it has associated the most with the week to relative or week relative to HCT. And so if I come over here and click on that, I can see which feature it has identified as the most relevant. In this case, it's the Enterococcus. Some of the others that show up here are the Clostridium, Lachnospiraceae. And so here, like in particular, you can see like it does look like these declined, and then there's some recovery after the treatment. You can also select these by clicking on these bars down here. And then the legend at the top of the figure is also going to tell you what is plotted on the x-axis or on the y-axis. And so you can sort through these from the features that were identified as the most important to the least important. And you can see that this plot, again, this is relative to the stem cell transplant, but we are seeing that a lot of these have decreases in abundance and then some recovery. We can also, of course, just like we could before, we can change how we're grouping these. And so, for example, if we switch to auto FMT group, we can see how these are changing over time. When grouped by the treatment versus control. And so, for example, in this one, the Ruminococcus, it looks like maybe there's some separation there. But again, that looks pretty small. And so really what we would want to end up doing is working with the LME, the linear mixed effects models, to try and see if there are significant changes in this taxon with time and with treatment versus control. You could provide these data to LMEs as well, so you could provide the bacterial genera to the LME to build those models. We're not going to do that right now, but something we could cover in the Q&A if there's interest. Okay, so the last thing that we're going to do in this section of the tutorial is, or in this section of the workshop, is we're going to generate a feature volatility plot. So same thing we just did, except we'll do this based on week relative to FMT. And so now the way that I did that before is I selected rerun this job. And so if I again select rerun this job, it will auto-populate with the data that I previously provided. And so sample metadata, our 4 artifacts as metadata, and then what we want to change here is the state column. And so we're going to do week relative to FMT. And we're going to execute that. So that is running now. You can see, just like before, this is going to produce 5 outputs. If I look over here, the one that I'm primarily interested in is the volatility plot. Okay, great. So mine finished running. So I'm going to take a look at that volatility plot. I'm going to view at Chime 2 View. And you can see that again, we are plotting based on patient ID as the group. And it is going to be plotting one specific bacterial genus based on what it has identified as being the most important. And so here in this case, it is identifying the lactobacillus as the most important. To me, I don't really see anything all that interesting showing up there. If we plot some of these different groups, we can see, like some of these things are things that are decreasing in abundance, and so there is some association with time here as we see these decrease in abundance prior to the FMT. But so far, I'm not seeing all that much that is very, very interesting. Here we're seeing some changes, increases in abundance, in some cases post-FMT. And so this is an unnamed genus in the Lactospiraceae family. And so if I go ahead and change to auto FMT group here, you can see some changes, but again, nothing that really looks very striking. Here we do see, so I guess the lactobacillus, what it identified as the most important feature. We are seeing some increases in abundance post FMT. Not a whole lot. Remember that when we come out here, we have very few samples left, and so this is not a particularly interesting region of this plot. But what I suspect is happening is this is a very low abundance feature, and so we're losing some of the signal in just the small area that this is plotted in this figure. If we hover down here, you can see some stats regarding this particular group. And so, for example, you can see that the global mean, so this would be the average abundance across all of the samples, is around 3%. Global median, quite a bit lower than that. Standard deviation, about 10%. And so you can see this is a low abundance feature that we're working with here. That the difficulty of seeing that might also suggest why we want to do some of the filtering filtering of low abundance organisms before generating these plots, just helpful for the visualization. Now there's one other plot or one other visualization that I want to look at here, and that is this accuracy results.qzv. And so for me, that's showing up just above that result. And so this is going to be showing just a summary of the ability of the supervised learning classifier that was used behind the scenes in generating this plot for predicting the time. And so what this is using is a random forest regressor. And so this is trying to predict a continuous variable based on the different genera that are provided as input. And so what you can see here is that we do get a reasonably accurate classifier. And so our squared value of 0.44, significant p-value here. And so what this is saying is that it is possible to predict time based on these combined genera abundances. And so that does suggest that it is useful to look at that volatility, that feature volatility plot. If instead we saw a high p-value here, say 0.5, 0.7, so something that was not in the significant range, that would tell you that we were not able to predict weak relative to FMT from the data that were provided as input. And so there's really no value in looking at the important genera there. Now this p-value is sort of an omnibus p-value looking across all of the different bacterial genera that are included in this analysis. And so while it tells you that there may be some interesting signal in there, it's not telling you about any one particular genus in there. Again, you would want to refer to the linear mixed effect models to try and determine if you have significant associations with any particular genus. That would also allow you to integrate these covariates in there, such as what group the individuals are in. Okay, so that wraps up the longitudinal microbiome analysis tutorial. Thanks for your time.